Good morning everyone, you are watching Kishore Manch and I am Rahul. Welcome to the first session of 6th August 2020 and today we are going to talk about Heredity and Evolution Part 6. Earlier we have seen 5 sessions for the same topic, so today is the conclusion of it. So please recall all your sessions which you have learned and seen on Kishore Manch, and we have Dr. C.V. Shimre in the studio. Thank you very much for joining with us, and we'll begin with the numbers of toll free, or with the help of them you can easily connect it with us. 1-800-111-265 and another number is 1-800-111-265 and the next number is 1-800-112-199, and you can also send us an email ciet.kishoremanch@gmail.com and all your queries will be answered smoothly. You can also watch this live webcast on NCERT's official YouTube channel. So let us begin with Heredity and Evolution with some recap. Ms. Shimre, Dr. Shimre, please. Now over to you.
Thank you, Rahul.
Okay.
So let us begin with some recap of the previous 5th part. That means that we already had, before that we already had 4 sessions, but we are not going to go back for now. So we'll just go back at this time to the previous session which that was held last week. So if we just go back to that slide, so we discussed about how evolution helps us in classification. If you can see the slide there, so it tells us that based on certain characteristics or features which are common in the organisms, we are able to classify them. So evolution, organisms evolve over time, population evolves over time. So then after that, having that idea about evolution, you're able to classify those organisms based on their similarities and differences or some shared characters. For example, as you can see there, presence of nucleus, or whether the organisms are unicellular or multicellular, or whether they are able to photosynthesize, or different kinds of characters that you observe in different organisms. So based on the similarities and differences, you are able to classify them. So that's what evolution helps us in doing. Then we also discuss about how can we trace evolutionary relationships. That means that how do we know a few organisms are related or closely related or more closely related, and some organisms are very different from each other. So how do we know that? So we discuss about, as you can see in the slide, there are certain ways of looking at it. Direct observation, we have been able to see, we discuss about some of the changes that is visible right now. For example, the beak length, you know, the shape of the beak, how the shape of the beak in different insects or different birds appear differently just because the food sources are different. Okay. Then we also studied, learned about the mosquitoes which have over time evolved in getting itself resistant to DDT, because we used to use a lot of DDT to control mosquito breeding population. So okay, now because of natural selection, so now they're able to adapt to that. That means they are now becoming resistant to that. There are other examples that we also studied. For example, how did we have so many strains of virus, common cold virus? So it is again one of those ways by which natural selection acted upon them, because of which we are having so many different kinds of strains now. So those are some examples that we discussed last time. Other examples you have seen, that you can go back to your slide. Then we also saw how humans have manipulated evolution, in the sense that yes, we have picked and choose the characters that we wanted. For example, as you can see in this slide, how did we manipulate the wild cabbage, and now we have so many different varieties yes, of vegetables coming out from that single wild variety of cabbage. So you can see there different cauliflower, you can see the cabbage, broccoli, red cabbage, broccoli, yes, and all those things, kale and that. So this how we are actually seeing the changes that has happened over time. But all these plant vegetables are still around us, you know, we can actually see them. So we know that these are related, and these have come from a single parent wild species of wild cabbage. Then we also saw okay, that there are certain other ways where you can trace the evolutionary relationships by looking at the organs which we call homologous organs. These are organs which have similar basic characters, basic structures. So as we took the example of this, in this example, we saw that human beings, yes, then the cat, then you have the whale, you have the bat, they have the similar basic structure. Yes. Because they have this similar basic structure, we can say that somehow these organisms, these groups of animals are somehow closely related, because they still share the similar basic structure. Then there is another thing that we discuss about. analogous organs. So this do not directly help in evolution at this time to trace evolutionary history, but somehow at some point they also indicate us that okay, some organisms evolve in certain place and they share certain similar characteristics. For example, you'll see that some organisms evolve at the same place because they share the similar environment. For example, for flying, all organisms which wanted to fly or which eventually were able to fly, that means because it was to fly, and so they had shared the similar basic structure again. Although the example that we are citing here in this one, it doesn't tell us anything much about evolution, but still it tells us that organisms also have similar characteristics based on the usage, because they want to fly, so they have like wings to fly. Birds have wings to fly, as similarly butterflies also have wings to fly, although they are not in terms of evolutionary history they are not related, but there are other animals or organisms which shows these kinds of characteristics, similar characteristics, because they share the similar environment. So that's what we studied. Then another very important way of tracing the evolutionary history was fossil. That was
yes.
what we discussed. So fossils tells us a lot about evolutionary history because we are able to date how old a fossil is using the radioactive dating techniques. So there are different techniques that are being used by scientists to trace the evolutionary history. I mean, how old these fossils are. So you're able to understand how old they are based on those radioactive dating. So then now you're able to see if they are very old, that means that they have been evolved much earlier. If their age turns out to be younger, that means that the evolution is much more recent. So that's how you're able to trace. For more recent organisms, you can actually use DNA to trace the evolutionary relationships, but DNA can give you as much very more recent things, because the very old fossils do not have DNA.
Yes, in the last session you told me that DNA can be, find some recent changes and
yes,
evolutionary things, but the thing is fossil can give you some deeper knowledge.
Right, right. So if fossils can give you very, I mean, it can be very old and you can still date the age of that fossil, but for DNA you cannot go that far back. So that's the advantage of using fossil. But definitely for more recent organisms, you can always use DNA, and that will give you a very precise result, So that there is an advantage in DNA also for the more recent organisms, but for the older fossils or the older organisms that existed in the past, fossils are more useful in terms of identifying how old they are. So that was what we discussed last time. So those are some of the fossils there. So today we will discuss some more about, for example, evolution by stages, how evolution happens. Evolution do not just happen at once, it's not like, okay, this group of animals did not have wings and now suddenly the next group of animals have wings and they're able to fly. It doesn't happen that way. As you can see in the slide, if changes happen bit by bit.
Okay.
For example, evolution of eye, it was not like, okay, the group of organisms suddenly appeared with perfect eye. It doesn't happen that way. So it was gradual. Over time, there was changes bit by bit in the development of eye, and ultimately we have so many different kinds of eyes. For example, insects have different kinds of eyes. We have different kinds of eyes. Animals have different kinds of eyes. But then we have developed over time. So it happens bit by bit. Such changes happens bit by bit. And it is not necessary that change that has happened at one point will be for the same purpose over time. For example, as you can see there, feathers in dinosaurs were present initially, but that was not for flying. That was for insulation in cold weather. But over the years, over the centuries, over the millennium, over thousands and thousands of years, we know that birds eventually evolved, and birds actually use these feathers for flying.
Yes.
Whereas dinosaurs use these feathers that was present in them to keep themselves warm for insulation purpose. So what I'm trying to say is that it is not necessary that one particular trait or character that was there in a group of species or organism will not be used for the same purpose over time. So the purpose of that character itself can change in different organisms. So that's what we saw. Then there is another property that we saw. So what is important here is that whatever it is, if they share similar characters, that means that they are more closely related.
Yes.
The more characters they share with each other, the more closely related are. That's what we can conclude from tracing the evolutionary history. So that's we said. Then let's come back to that again. What is evolution? When we say evolution, some people think that, okay, evolution means we become better and better, or it's progress, which is not so, you know. Evolution just means that there is change, and change to adapt to the situation, you know. And so whatever existed in the past, it was good for that time.
Okay.
Now we have different kinds of species or organisms existing, and it is good for that time, it is suiting for that time.
Right.
So things will change again over years, in thousands of years time, or like million years of times after now, we might see totally different organisms again, you know. So it doesn't say that it is progress. It doesn't mean that it is progress. It just means that there is change happening because of various other reasons. It could be advantageous in many cases, but it could also not be advantageous, as we saw in the previous sessions about genetic drift, where changes were not it was by chance and they did not have much of advantages. But then eventually, if they're not advantageous, they will slowly be eliminated from the population. So that's what naturally happens. I mean, that's what we observe in nature. But again, coming back to that, evolution does not mean progress.
Okay.
Right? So again, another very important point is evolution do not happen linearly.
Okay.
That means that, for example, we all still hear people saying, you know, that human beings evolved from chimpanzees.
Yes.
That's what you always get to hear,
Yes,
even
yes,
today, you know.
yes.
It's so, so that's not something that happens. It doesn't happen that way.
When we see monkey or chimpanzee, they say that we
have evolved from those.
Yeah, evolved from those.
Yeah, so, but it doesn't happen. Evolution doesn't happen that way. So it is not linear that, okay, from one species it has become this, and from this
Yes,
species it
yes,
has become this. It doesn't happen that way. So it has multiple branches. It can have multiple branches. It is quite possible that human beings and chimpanzees have shared some common ancestors in the past.
Okay.
That means that we might have evolved, or we could have high chances that we could have evolved from some common ancestry. We had some common ancestors, and from that we branch out into different organisms. We branch out as human beings, and chimpanzees eventually became chimpanzees. But we did not evolve from chimpanzees. So it will, students should be very clear about that. That evolution do not happen linearly.
Okay.
So it happens in branches. It branches out to different other organisms, different groups of animals. They might survive if they are adapted to that. They might not survive depending upon their ability to adapt, or changes in the genes that's happening in the organisms and in the populations. So that's what. Another important character is that, okay, fine, evolution is not progress, but of course we see more and more complex body designs evolve over time. For example, initially when life started evolving, it was pretty simple. But now we see more variety, more complex forms of characters in the different organisms. So we can say, at best we can say that when we say evolution, we can say that more and more complex features have evolved over time. But it is not progress, as we said. So it is not better as such. It is not, we are not developed. We should not consider ourselves more, you know, higher than any other organism. It just happens that we existed at this time, and we might not exist in the future. So we might change, and so as different organisms can change over times, or over the years, or over different centuries, you know. So that's what we studied. Let's come back to human evolution. Let's end this chapter with this topic, human evolution.
Okay.
So as we all know, evolution in human beings can be traced back to some place in Africa.
Yes.
So from there, we started spreading out. We started migrating to different places. As you can see, Africa, as you can look at the climatic conditions there, it's hot, warm there. And so you can see people with different features, you know. You can see people with different skin color. You can see the textures of the skin, or even the facial features, and all those different features you can see typical of people living in Africa. But then if you now look at human beings living in different places. For example, Rahul, you and me, we look very different, right?
Yes.
So it has something to do with the environment that we have been living in, you know.
Yes.
So this is what happens. For example, the white people, they look very different from us.
Yes.
So again, so they are very different from African people, black African people.
Yes.
So although we initially human beings evolved from Africa, I mean they were traced back to Africa. Now if you look at different people in different countries, we look so different. And so this all about so how changes have taken place over the years, you know, to suit ourselves to the climatic conditions. For example, white people did not need so much of melanin, and so now they have white skin, you know. So things happen over time.
In short,
over
we can
ages.
say it takes thousands of years, hundreds of years to see the evolution and to read about it.
Yes, it
and to
takes
feel the difference.
Yes, it takes years and years, thousands and thousands of years to see those changes happening actually. So again, when we say evolution, it's not like okay, something has changed immediately from here to here and you're able to pinpoint it, okay, here evolution has happened. You cannot do that in case of evolution. It's not possible to pinpoint that okay, from this point to this point we have seen evolution. It doesn't happen that way.
So
you
it
know?
is a kind of
so
research.
it's a small changes that's happening over time. At some point the changes are so drastic that now we are able to see. But if you trace the evolutionary history, if you actually had the DNA, if you had it over times, over the centuries, over thousands of years, then you might be able to see exactly point out, okay, from this point this much changes has happened. But since we do not have so much detail about the past, all we have is fossils, you know. Fossils can at the most tell us about the age of the organism and the shapes, of course, the traces it leaves behind. You can find that. So it's still, you know, it's still not very convenient to trace exactly what happened in the past using fossils, but it can actually help us in giving us some sense about okay, this were older than these organisms, these groups of organisms evolved far earlier with this type of characteristics. And so since these organisms have these characteristics and these organisms have this much of age in terms of carbon dating or any other technique of dating the age of the fossil, so we're able to put them in place, you know, that okay, you can arrange this way that maybe this has happened in the past. So that's how we try to predict the past using fossils.
Right.
So that's where we end this chapter. As Rahulji pointed out in the beginning, that we already had 5 sessions before. So what we are going to do now is go back to all those 5 sessions that we had done and try to.
This is the wonderful thing, so that you can memorize all the sessions and the complete detail of this topic, especially.
Yes,
so it will be
that
really helpful.
Yes, so that it will tell us exactly what we have done over the past 5 sessions and 6 sessions today, including today. So we, if students have not seen the past 5 sessions, they might want to go back and see the past 5 sessions in this YouTube channel again. So they are all there. In the title will be Heredity and Evolution Part 1, Part 2, Part 3, Part 4 and Part 5. Okay. So even if you have not seen but learn heredity and evolution at some point, at some place, then you should be able to make use of this recap. So it will be mostly beneficial for those people who have learned about heredity and evolution in the past, or who have attended the previous 5 sessions. So this recap is especially for those students. So let us begin. So how did we start? Can we go back to the slides, please? So can you please take me to the slides? So how did we start? We started with the definition of heredity. What is heredity? It's a transmission of traits from one generation to the next generation. So it is also called inheritance. We mention about inheritance. We talk all the time in our daily life. Who is going to inherit the property of our parents? You know, we use that all the time. So similarly, what are we going to inherit our next generation, our next offspring? You know, parents inherit their genes to their offspring. So it's the same thing. So in heredity, the parents inherit their genes to their offspring. So that's what we study. That's a definition of heredity. Then we also said that the more closer they are, siblings looks more closely similar. The farther you are in your relationships, the more dissimilar you're going to look.
Okay.
That's what it is. These two are siblings, and so you can see how similar they look, you know. But it's not necessary that all the siblings are going to look
Yes.
exactly the same way.
Yes.
Some siblings look very different, and there are reasons for that. But the more closely you are related in blood, the more similar you are going to look.
Yeah, there is no compulsion for siblings
yes.
that they look, you know,
similar, but
together.
many a times
Most
it
of the
happens
times
that
people do such things
yes.
in the parties and
Yeah.
when they meet together. So this is really a strange thing, and
Yeah.
you should learn from science. But that this is not a mandatory thing.
Yeah, it's not necessary that they should look
Yes.
similar, but it turns out that many a times they look similar. They just happen to look similar because they share genes from a same parents, you know.
Yes.
So that's how it happens. But why are those these similarities appearing? Because there is something called sexual reproduction in human beings and other animals and other organisms, where there is exchange of genes happening in the during formation of gametes and all that. And so kids or the offsprings inherit different forms of genes from their parents, and so because of which they look not dissimilar from their other siblings. So that's how you see the variations in different individuals. Then we also define what trait is. So what is trait? Trait or characters, which we call, that means what you see in me or what you see in yourself. So these are genetically determined. That means 22 pairs of autosomes and 2 sex chromosomes. 22 pairs of autosomes and 2 sex chromosomes. That means the 2 sex chromosomes are X sex chromosome or Y sex chromosome. So that's what we have in human beings. So different organisms have different numbers of chromosomes. So it occurs in pairs, which we call homologous chromosomes, the pairs. So that's another way of seeing that chromosomes. So every chromosome have different genes. I mean, so many thousands of genes. In human beings, we say that there are about 25,000 to 35,000. So depending upon the research findings, the number varies. So, but of course, there are many genes out there. So between 25 to 35,000 genes that's present in different chromosomes in different organisms. This is the X chromosome. As you can see there in the picture, it has 1,098 genes, you know.
Oh my
so
goodness.
in just one chromosome. And so 25,000 to 35,000 chromosome is spread over those 46 chromosomes.
Do I need to zoom this picture for the
No,
kids?
no, that's just for information. No,
Okay.
they don't have to see that. So then from where do we get these genes, you know? Since genes are responsible for the inheritance of these traits, where do we get these genes from? We get these genes from our parents, as we discussed in the beginning. So from our parents, fine, but from where, which part of it? So our parents, they produce, or any organism, they produce gametes for reproduction, for the purpose of reproduction. So they produce gametes. For females, it is a egg or the ovum, and for males it is the sperms. And so these sperms and ovum are the ones which are carrying those genes, which are carrying those units of inheritance, which we call genes. And so whatever genes are present, whatever is present in the somatic cells, that has nothing to do with inheritance. Only those characters which are present in the changes or anything that's happening in the gametes, that is a sex chromosome, that is the sperms and the eggs, those will be inherited to the offspring. So offsprings inherit genes from the parents through the gametes. The gametes, male and female gametes, as students have studied in the past, that there is something happening, that the process called fertilization. During fertilization, fusion of these gametes happen, and after the fusion, zygote is produced. From the zygote, the cell keeps on multiplying. Ultimately, you form the full-fledged organism. For example, from the zygote, we also develop from zygote.
So
Any organism.
So this is the hierarchy of reproduction.
Yes, that's how reproduction happens. So reproduction starts with formation of gametes. Then the gametes fuse. After the gametes fuse, they form a zygote. The zygote divides continuously. Ultimately, you find the full-fledged organism. So that's how it happens. Then something that we discuss about, so haploid and diploid cells. So what are these haploid cells? As I mentioned, homologous chromosomes. Chromosomes exist in pairs. So that means if you take half of those pair, it will form one set. So you represent the S, small letter N, as you can see in that slide. So when they have two such sets, for example, the 46 chromosome represent diploid cell. cells which have 46 chromosome, that means 2 sets of chromosomes, they are called diploid cells. If the cells have only 1 set of chromosome, which is represented by a small letter n, then you call that such cells as haploid cells. For example, gametes are haploid. And that is the reason when 2 gametes fuse, that is the male gamete and the female gamete fuse, they form a diploid cell again. So if the haploid also had 2 sets of chromosome, then what will happen is after fusion then we'll have 4 sets of chromosome. That means that is not possible. And so in order to retain the chromosome number, haploid cells have 1 set of chromosome and 2 haploid cells. That means the gametes, when they fuse, ultimately you have the diploid cell back again. Okay. So that's how it happens. So let's go to the homologous chromosomes. And what are alleles, you know?
Yes.
So what are these alleles? Because we have been talking about alleles, so let us recap again. So as we said, they are genes. Genes can be present in different forms, different versions.
Okay.
So when genes, the different versions of these genes are called alleles. So as you can, I'll just show you a picture of this. You can see in this is a homologous chromosome. Look at the picture carefully. That is a homologous chromosome where you can see a pair of chromosome there. And you can see a gene there. As you can see there is a gene there for the color of the flower. This represents a chromosome of, for example, let's just say a chromosome from a pea plant. So it is a homologous chromosome from a pea plant. And one of those, as you can see, the band there, it represents a gene for color, flower color. But as you can see the label there in the top, you can see allele for purple flowers, and in bottom you see the allele for white flowers. That means they are the same gene which is for the flower color, but they represent different flower color. That means so they are in different versions now. The genes are in different versions because one of those genes represent purple flower color, another gene that is below that represents white flower, and so we call those 2 versions as alleles, you know. So they should not confuse with genes and alleles. So in this case, as it is very clear in the picture, those 2 bands represent the gene for flower color, but the genes represent different flower colors, so you call it alleles.
Okay.
So the different versions of the genes are called alleles. So that's very important. So let's come back to alleles. So the 2 alternate versions or the forms of genes are called alleles. In homozygous condition, you will find that both the alleles are same, but in heterozygous condition, the alleles are dissimilar. For example, in this case, the alleles are different.
Yes.
One is for the purple flower color, another one is for the white flower color, and so you call it, they are in heterozygous condition.
Okay.
So if, for example, both the chromosome had, or both the genes had allele for the same flower color, then you will call them homozygous, in the chromosomes are in homozygous condition. But since in this case it is different, you call it, they are in heterozygous condition. So in nature what you will see is that one of these allele, now that you have 2 types of alleles there, so what will the flower look like? Now you have the gene or the allele for purple flower color and the allele for white flower color. So what will the individual exhibit? You cannot have both purple and white flower color. You have to have only one.
Okay.
You know. So in Mendel had already found out what's going to happen, and he found out that purple color, as long as there is allele for purple flower color, the plant will have purple flower color. So any plant with this kind of genetic constitution, with this kind of chromosomes, with 2 versions of the alleles, then always one of those allele will be expressed, will be seen.
Okay.
The flower color will always be purple in case of pea plant. That's what Mendel already found out. So what is exhibited in case of heterozygous condition, he called it dominant allele. That means in this case, since the plant is going to produce purple flower color, we call it the purple allele for the purple flower color is dominant over the allele for the white flower color.
Yes.
So that's how you define dominant and recessive allele. So you call those which are not expressed, the alleles or the genes which are not expressed, for example, in this case, the white flower color, you'll call that a recessive gene. So that's how you name them. so Mendel found out through his experiments that certain characters are always dominant over another character. For example, the flower color, in case of flower color, it is a purple which is always dominant. So when in a heterozygous condition, you will always have purple flower color, you will always have axial flower, presence of flower in that. You will always have yellow flower, yellow seed if in a heterozygous condition. So similarly, he found out for 7 different contrasting traits. So that's what he did. So let's go further. Then there's something called genotype and phenotype. So what's all this genotype and phenotype? Genotype is a genetic constitution of an organism. Phenotype is a visible expression. So whatever you are seeing is the phenotype, which can be different from the genetic constitution. For example, in the flower color which we just saw, there was gene for the white flower color, but still the plant will produce only purple flower color. So the phenotype will be purple flower color, but still the genotype will contain the white flower color genes. Okay. Then we also studied about something to do with self-pollination, cross-pollination and true breeding, which are very important. So I'll not get into the detail of this. So then Mendel did some interesting experiments. So he crossed organisms, he crossed different individuals with different characteristics. So here, for example, he did the monohybrid cross. What is this monohybrid cross? It is a cross between individuals heterozygous for one character. That means, as you saw, what is heterozygous condition? So one character heterozygous, for example, the one that we saw, it is heterozygous for one character. That means the flower color, it was heterozygous for the flower color. That means we had genes for purple flower color as well as genes for white flower color. Then he also did an experiment crossing individuals heterozygous for 2 different characteristics, which we call it dihybrid cross. Okay. Then there are trihybrid cross. So several experiments Mendel did. He did all kinds of experiments because he was interested in science and he was good in maths also. He did a lot of calculations. So what happens if I cross this individual with that individual? What happens if I take these 2 characteristics? Whether another characteristic has influence over another characteristics during the times of inheritance, or when the genes are transmitted, or when the gametes are transmitted. So he did all kinds of experiments. And so he predicted, he tried to predict some of those characteristics. For example, in a monohybrid cross, so he tried to calculate and he tried to find out. So what's going to happen if he tried to cross this monohybrids? For example, as you can see in that slide, there. So if you cross, look at the F1 generation there, purple flower color there. So you can see the slide there, please. Yes. No, there. So if you can see. No, you have deleted. I think you can just go back, undo that. Yes, I think Control Z.
Oh, sorry.
So anyway, so this is the slide. So if you cross the hybrids with which are in heterozygous condition, so he predicted what's going to happen. So as you can see in the offsprings, what kind of offsprings those organisms, those crossing is going to give rise to. You can see there are 3 purple flower color and 1 white flower color. So you can say the phenotypic ratio, he came up with the phenotypic ratio of 3:1. So, but if you look at the genotypic ratio, you'll find that it is 1:2:1. So the genotypes, as you can see there, the big P's and the small P's there, if you start counting them, then you'll be able to find the genotypic ratio and the phenotypic ratio. So Mendel did a lot of such calculations. So he calculated those using Punnett square, but he also actually planted those individuals. So when he planted the individuals in the field experimentally, so he had to take those plants, those hybrids, and he had to actually cross them manually, because if he doesn't do those crossing manually, fertilization manually or pollination manually, what's going to happen is some insects or some other birds or any other organisms, any agent can do the pollination, and it might bring in the gametes that he did not want. And so the situation has to be really controlled. He has to make sure that the pollination between the individuals of his interests is pollinated, and so he had to actually manually pollinate all those individuals of his interest. And so when he actually pollinated and he grew all those individuals with the characteristics that he wanted to see, he found the result in the field similar to what he calculated using the Punnett square. So the predictions almost came out to be same with with what happened in the field actually when he grew the plants. So that's something very interesting, but it's not exactly. For example, as you can see in this slide, you can see the ratio there. We say that 3:1 is the ratio, phenotypic ratio, but as you can see in there, the ratio is 3.15:1, right?
Yes.
So it is not exactly 3:1. The prediction through calculation in theory was 3:1, but when he actually grew those plants, he found it to be, that means that 3.15:1 was the ratio that he came with. So he had to plant them, he had to see what kind of flowers came out of those pollination. So he had to pollinate them, he had to see what kind of plants, seeds were produced, he had to plant those seeds again and see what kind of flowers were produced, and so that's how he counted the plants and the flowers, and he came up with this ratio.
Okay.
Similarly, he did with that with the dihybrid cross also. So he took two different contrasting characters. For example, in this case, as you can see in the slide, the seed shape and the seed color, where he took those two characters. He wanted to see whether the seed color had any influence over the inheritance of the seed shape. So that's what he tried to see, and he found out that it had nothing to do with, these two characters had nothing to do with each other during the formation of the gametes. That's what he came up with. That was a finding that he came up with using this dihybrid cross. So that was about dihybrid cross. So what he found was that, okay, changes happen, but you look at the allele frequency, we have not found anything, any new species. It is just that, okay, some plants in some generations, some plants had more of these characteristics. In another generation, some plants had more of these characters. So characters keep on moving in the population. Sometimes you have this green color seeds more, sometimes you have yellow color seeds more, depending upon what kind of pollination was happening in the field.
Okay.
But things did not really change in that, you know. Evolution has really not happened. It was just that there was intermixing between population were intermixing between the inheritance happening, but then you do not really see a new species originating from that. Then in between, we also studied about this sex determination. So then in human beings, how the sex is determined, we came across this. So we always hear people say, oh, who is responsible for the sex, you know. So this is a very wrong term that we are using. So nobody is responsible as such. It just happens that male gametes can have the X sex chromosome or the Y sex chromosome,
okay,
whereas the female gamete can have only the X chromosome. So that's what happens. So it is, nobody is responsible. It is just that the gametes can have either of those X or Y chromosome in males, whereas in female it can have. So whatever is, fertilization is happening with the female gamete, whether the sperm contains the X chromosome or the Y chromosome. So that's how you have the male child or the female child. So we should always avoid fixing responsibility on who is responsible for the sex of a child. So that's something which is very important for students to know, so that they can educate other people as well, that nobody is responsible. It is just by chance it is happening. So then we came to evolution. So we talked about evolution. So what is this evolution? These are heritable genes. Then what are these? Another way of defining evolution is changes in the allele frequency. As we said, in Mendel's experiments, he saw all those kinds of what kind of traits will be inherited and all those things. But evolution, we did not really talk about evolution. Allele frequency changes did not happen. Okay. So in order to evolution to happen, allele frequency has to change. The number of alleles, for example, we saw the allele for the white flower color or the purple flower color. Yes. So all those things has to change in the population. Only when there is drastic change in the allele frequency, then you'll say that there has been evolution. So that's what we saw. We talked about microevolution. When changes are happening, the changes in the allele frequency are happening within the species, then we call that microevolution. When it is happening in a huge, the changes are happening in a very broad way, then you call it macroevolution. Okay. For example, yes, evolution of mammals, birds, and so on. So that's what we discussed. Then we also calculated the allele frequency, how important the allele frequency is, how it changes the population. So how allele frequency can be changed, we discussed about that. It can be changed by mutation, genetic recombination, non-random mating, natural selection, gene flow, genetic drift. So these are mechanisms through which allele frequency can change. And so when the changes are so drastic that individuals in the population or species of the same species but in the different populations, if they are not able to breed amongst themselves and produce viable offspring, then we say that speciation has happened or different species has been formed. So that's how we say when they are not able to interbreed. So when allele frequency changes become so drastic that the individuals are not able to breed amongst themselves and produce viable offspring, then we say that species formation has happened, speciation has happened. So that's what we did. Then we discussed a little bit about Charles Darwin, what he did with his experiments and all that. So we talked about in detail about the natural selection, how it is advantageous for the organisms, whereas in case of genetic drift, it has nothing to do with advantage. It did not necessarily that the characters will be advantageous. That's what we discussed. Then we also discuss about in detail about founder's effect and bottleneck effect, which students can go back and see the videos for that. So these are changes which might be advantageous or which might not be advantageous, unlike natural selection. So that's what we studied. Then we studied about gene flow, then speciation that I just described. So those are the things that we came back to this evolution and classification, how evolution helps us in classification. Then we today we discuss about tracing evolutionary history. Then we talked about all that. But what is, at the end, why are we studying all of this? You know, it's very important. Let me give you this in 1 minute. So why are we even studying heredity and evolution? First of all, a lot of medical uses. In agriculture, how much we're using. We just discussed about one example, how from wild cabbage we got so many different varieties of vegetables which we like, you know, we enjoy different varieties. So that is one way. And you can now, because of genetic engineering, you can do a lot of those changes in the genes, you know, so that you can get genes of your interest, organisms or species of your interest, which will be pest resistant, which will be drought resistant, which will be flood resistant, all kinds of things, organisms you can now have because of genetic engineering. So unless you knew the evolutionary significance or unless you knew the genes of such organisms, you will not be able to do all this, you know. Look at the medicines, the changes that has, all the progress or development that has happened in the field of medicine. So we have been able to come up with different vaccines, different medicine, different drugs, all because we understood the genes and the genetics and the evolutionary significance of all of those, you know. And evolution also tells us how we are related to some organisms and how we are not related. So it tells us about the diversity, how the diversity that we see today came into being. So it tells us a lot about all this, and it tells us how to apply those knowledge in our daily life as a scientist, as a researcher, as a farmer, you know, as an individual. How can you avoid certain diseases, or how can you go for genetic counseling, which we did discuss in one of those sessions. So there's so many things that we can learn from this chapter. So students might want to go back and look at the previous 5 sessions and try to understand holistically what this chapter is all about. Right?
Yes, you should spend some time so that you may have the thorough knowledge of each and everything of heredity and evolution, as this part 6 is concluded now with all the revision material of part 1, 2, 3, 4, 5. Thank you very much, Dr. C.V. Shimre. You have described very well, and this is a part of research, as we are getting so many results, and this is the contribution of ages. I think the people who are doing researches and you know, giving the results for us, and those data is, you know, stored for us. So we are getting the results in terms of medicines, in terms of genes and everything. We are in the field of agriculture. There are so many things I have learned, and now I can say one thing with the confidence: we are not evolved from chimpanzees. Thank you very much for letting me know and enhancing my knowledge. Thank you very much for joining with us.
Thank you.
So that's all for this session. Please be with us. Our next session will be commencing very soon. Thank you very much. Namaskar.